Just welcome Ali, who's came to Melbourne 3.5 years ago from Iran, where he got very excellent grades in his studies in statistics and one of the top marks in a national competition for entry into master's programs. And you'll see from his talk today that he's shown up to this promise and has done some really good work that I hope you'll enjoy. So at this point, I'll pass over to Ali. Let me just say, no oral, please everyone mute as usual, and probably also best to turn the video off. If there is something during the talk, feel free to put questions in the chat. And if they're immediate ones, I might, decide to pose them. But in general, we'll wait to the end for questions, and then you can either put them in the chat at the end, or you can ask them orally. We'll come to that point. But anyway, let's go ahead, and thank you, Ali. Please go ahead.
Thank you, David, for that kind introduction. And thanks everyone for attending my PhD completion seminar. Today, I'm going to talk about inference under the Coalescent with the combination, and I'll introduce my method, ARGinfer, that infers the ancestral recombination graph. So let's first give an introduction to the Coalescent with recombination. It is a stochastic process that describes the evolutionary history of a sample of DNA sequences backwards in time. A realization of this graph is, this process is a graph called Ancestral Recombination Graph, or in brief, ARG. So let's explain the process here. So this is an example of an ARG for 3 sequences, A, B, and C, from the current generation. So according to, based on the Coalescent with the combination, going backward in time, the time to the next event is exponentially distributed, and each branch can experience either a recombination or a coalescence event. For example, here, the first event at time T1 is a recombination event on branch A. So I labeled them, I labeled the nodes as the branch above. So this recombination splits the branch A to 2 new branches. One carries the ancestral material right to the breakpoint Z, and the other carries the ancestral material left to the breakpoint, this branch here. And going back in time, again, a new time exponentially distributed, T2, the event is a recombination event again. And the third one at T3 is a coalescence event. So what happened in coalescent event is it merges 2 sequences. For example, here, this sequence and this sequence, they merge and coalesce, resulting in one parent sequence we see here. So this process continues until we have only one branch left, or here. We call this as most recent common ancestor. And the time to this is called TMRCA, or time to the most recent common ancestor. So as we see from this graph, ARG includes a lot of information about the history of these 3 sequences. We have the ancestral sequences here. There's their ancestral material, the times of the events, the recombination event, the recombination breakpoint, and so on. And ARG also includes all the genealogical trees at each site. For example, for segment from 0 to X, we see this tree that suggests first A and B coalesce at T time T6, and then they find their most recent common ancestor at T8. Simply, we can extract other trees. For example, this is the tree for segment X2Z that says B and C first coalesce at T3, and then they find their MRCA at T8 again. And lastly, for the last segment from Z to 1, the topology is exactly the same as, not exactly, it's the same as the second tree, but the TMRCA we see is different, and it's a T7. So again, ARG includes all this information, but we don't have it in reality. In reality, we have only the sequences, the current generation. So if we could have the ARG, that would be very much luck for population genetics, because many inference problems would be simplified. For example, we could do recombination and mutation rates, and effective population sizes. We could infer them simply. Time to the most recent common ancestor, allele age, or demographic histories of population. But the problem is, it's very hard. It's difficult to infer ARG. It's been active research for over 25 years. And even the history of using MCMC goes back to early 2000, about 20 years ago, and still we don't have an efficient MCMC algorithm that that assumes the coalescent with the combination and infer ARG. The reason, or some of the reason, is because many different ARGs have a very similar likelihoods. So it's very hard to explore the space, and the space there are infinitely many compatible ARGs for with any given input data. So it's an infinite space. Because of these difficulties, so a major focus has been on development of approximation to the coalescent with the combination, one of which has gained a lot of interest by other researchers is sequentially Markov coalescent, that introduced by Gil McVean and Cardin in 2005. So the SMC approximates the coalescent with the combination by prohibiting trapped genetic material. So let's first explain what is trapped genetic material here on this ARG. So these two children, they coalesce, but they don't share any overlapping ancestral material. Therefore, the parent sequence has a gap in between the ancestral material. This gap is called trapped genetic material. And if a recombination occurs on this gap, we call the recombination as non-ancestral recombination. Otherwise, the recombination is ancestral. So now, what the SMC suggests is do not allow this type of events. So if we don't have this type of events, then the process results in a Markovian process that is Markov along the genome and reduces the ARG space. So many simulation methods have been developed using the SMC. Also, some inference methods have been developed. So now let's take a closer look at this and see what information we lose here. So as I explained, SMC does not include non-ancestral recombination because we don't have trapped genetic material there. For knowing how much of this recombination or this information in an ARG is non-ancestral, I've done a simulation study. I've simulated the ARG of 50 sequences with variance recombination and mutation rate. So here rho is a scaled recombination rate per sequence, and theta is scaled mutation rate per sequence. So for 40 to 1600. So in this plot, the x-axis is the logarithm of the number of SNPs, and y is the logarithm of number of recombination events. So this green line here is the ancestral recombination. This is what SMC and SMC-based methods can model. The red line is the non-ancestral recombination, and the yellow is total number of recombination. So as it is clear from here, the non-ancestral recombination approaches to the total number very quickly. Like for this area, we see that the majority of recombinations are non-ancestral. So this suggests that if we include this information, which is a huge amount of information in inference, we may have a more accurate inference method. A breakthrough in population genetics occurred in my opinion, in 2016, when Jerome Kelleher and colleagues in Oxford introduced this data structure, tree sequence, that offers great orders of magnitude efficiency gains in storing and simulating DNA sequences. So tree sequence stores genealogical and variation data very compactly. For example, for 10 billion simulated sequences, each with 100 million sites or base pairs, the common format VCF needs about 25 petabyte. But for tree sequence, it only needs 1 terabyte, which is about 25,000 smaller. Also, tree sequence permits efficient simulation of millions of human genomes under the Coalescent with recombination. It's something that wasn't possible before, and it is still orders of magnitude faster than the existing methods that are based on the SMC and other approximation methods. So this was the motivation of my research, of my thesis. And now let's see where this efficiency come from. Here I just explained the idea of TS, not the TS that what it includes. It includes 4 tables. I want to explain them here. So these 3 trees are the marginal trees in an ARG, one from 0 to X on the genome, and the other one from X to Z, and the last one from Z to 1. So we see here the colors show the identical branches. We see that branch A is identical in all the 3 trees. So the traditional format, we separate trees, store trees separately. So we need to store them 3 times. But what TS suggests is only store identical branches once. For example, A, we just store it once for the whole genome from 0 to 1. Again, for branch B, we see that it's identical for all the trees, so we only need to record it once. That's where the efficiency comes from. So the same group in Oxford uses tree sequence and developed TS Infer to construct an ARG. TS Infer can handle over 100,000 genome scale sequences, and they applied TS Infer to UK Biobank data, about 487,000 sequences, and it took about 3 hours to construct the tree sequence. It's very fast. And also it generates point estimates of some ARG properties, more precisely the tree topologies. But the TS Infer does not infer event ARGs, event times, although I know TS state is under development for inferring times, but still it's a point estimate and lacks measures of uncertainty, which we have another model with called ARGweaver, developed by Rasmussen in 2014. So ARGweaver is the state-of-the-art probabilistic model and is a substantial improvement over the existing probabilistic model and can handle about 50 sequences with nearly 2 million base pairs. ARGweaver assumes the SMC, therefore it doesn't model the non-ancestral or trapped genetic material, and uses a discrete time axis. So for the time, they first pre-specify the axis and then force the events to occur there. So now, this is our motivation question. So this question is what I aimed to answer in my thesis and in this talk. Can the TS help us to perform accurate probabilistic inference under the coalescent with the combination? So first, we realized actually TS is not an ARG. It does not include all the information we require for ARG inference. So we came up with a new idea and designed a new data structure, an augmented TS, retaining all its good properties to store all the information we need. That is, for example, we need recombination events and breakpoints, all the ancestral sequences, and so on. The data structure is called Augmented TS or ATS, and I'll explain after this slide. So this is the data, the input of the algorithm we have is a sample of DNA sequences. We assume that each side can mutate at most once, infinite size model of mutation. And also we assume the ancestral alleles are known. So with this assumption, we can recode the DNA sequences in 0 and 1. So here we have 3 sequences, A, B, and C, each with 4 SNPs, all of which are segregating. So this is not the case in reality, definitely. So we represent the data in our way. That is, for each SNP, we only record the sequence that carries the derived allele. For example, SNP1, sequence C only carries the derived allele, so we only record C. For SNP2, A and B are the sequences that carry the derived allele, and so on. So now the data structure. We have 3 sequences, A, B, and C. So I just labeled the branches. Branch A, so what we record is the name of the branch and the ancestral material it carries. For example, branch A carries ancestral material from site 1 to site 4, and S is the descendant samples. So A is a leaf, the descendant sample for A is A. And now from this, we can go check from the SNPs. We see that for SNP3, the sequence that carries the derived allele is A. That therefore, we can put the mutation for SNP3 on this branch. And similarly for the other 2, going backwards in time, there is a recombination on branch A, resulting in 2 branches, 1 and 2. 1 carries the ancestral material left to the breakpoint, only site 1, and 2 carries ancestral material right to the breakpoint from 2 to 4. Continuing, the next event is a coalescence between 1 and B. The parent branch is 3 and carries ancestral material from 1 to 4, and the descendant samples are A and B. So now we go to this data and check for SNP2, we have A and B. Therefore, we can put the mutation on this branch for SNP2. And this process continues. The other event is a coalescence between 2 and C. The parent is 4. A and C are the descendant. We check the data, and the SNP4 has only A and C as sequences that carry the derived allele. So we put this SNP, the mutation 4 on branch 4. And lastly, 3 and 4 coalesce, and the resulting parent is 5. We see that the descendant samples are A, B, and C. This suggests that this is the MRCA for all the segment, and the process is finished. So now with that, we developed an MCMC algorithm and assumed that the coalescent with the combination is the prior. In here, I only explained the key step of the MCMC algorithm, that is to define a proposal distribution to explore the ARG space. So we are dealing with a very complex graph, so it's not that easy to propose transitions that are reversible. We introduced 6 transition types that I'll explain here. The first transition type is subtree-pruning and regrafting, SPR. So on the left graph, so left graph is the current ARG of 3 sequences, each with 10 sites. To apply SPR on, first we randomly choose a branch. In here, we choose branch B. and then prune it from the arg. That is, we separate it from the parent here, resulting in a floating lineage, which is B. And now randomly choose one of the existing branches on the arg. In this example, we choose 5, and then simulate a time for the coalescence, T5, and then we rejoin B and 5, resulting in this parent branch. So on the right, we see the proposed arg after the SVR. The second transition is removing an existing recombination event. It is clear from the name. So we choose one recombination event at random and choose one of the parents and remove it. For example, here we choose parent 1 and remove. So the child follows the path by the other parent in here 2, resulting in this graph on the right. The third transition type is to add a recombination event. So one of the branches, again, at random is chosen. Here we choose branch B, and then simulate a breakpoint on branch B. Breakpoint is 3 here, and then split the branch to 2 new branches, 6 and 7. One of the parents follows the existing path at random, and the other parent, 6 here, will rejoin the arg using SPR. So here we 6 and 5 coalesce at time T5 with parent 8. The fourth transition type is resimulate a part of arg under the coalescent with the combination. So here we allow the pruned lineage to experience recombination. So first, we randomly choose a branch on the arg. Branch B is chosen, and then remove all the events that this branch experienced from its time to the root, to the GMRCA. So here we remove this recombination event, and then as a result of that, we remove this coalescence event. And again, as a result of this, we remove the coalescence event at time T5. And then this is the current state of the arg. So this green line means the recombination links that are ready for break for having recombination on. So going back in time, the first event that B experiences is a recombination resulting in 2 pruning parents, P and 8 and 9. And then so we continue the process and resimulate it under the coalescent with the combination until all the branches rejoin to the arg, resulting in this graph on the right as a proposed arg. The fifth transition type is to change a recombination breakpoint. So we choose a recombination. Here, the recombination and time T2 is chosen. And then we simulate a new breakpoint for the recombination. Here, the original breakpoint was 2, and now it's 8. So this seems a very small change on the R, but actually it can shuffle the MRCAs, like big changes on the R. Let's check here. So when we have B as breakpoint 8, then the ancestral material on 4 and 3 changes. That's a direct effect. But let's check 5. So for 5, we originally had ancestral material from 0 to 2 and 6 to 10. But now it has from 0 to 8, 0 to 6. And then there is an MRCA for 6 to 8 here. And then it has material from 8 to 10. Similarly, for 6, we have MRCA for 2 to 8 here. So even this small change will result in like big changes on the ancestral material and on all the MRCAs that need to be addressed. So all these transition types kind of deal with the topology. The sixth transition is to resimulate the event times under the coalescent with the combination. So we start with time 0 at the present, and going back in time, we resimulate all the times under the coalescent with the combination. So resulting in these times T'1 to T'5. Was it a question? Okay, I'll continue. So now that I've introduced all the transitions, I've implemented this MCMC algorithm in software ARGinfer. And here we assess the performance of ARGinfer using simulation data. So I've simulated 150 datasets using MS Prime for 10 sequences with scaled mutation rate 20 and mutation rate to recombination rate 1, 2, and 4. So this is for different scenarios for recombination rate. And then we run the algorithm for 2 million iterations and discarded 1 million of them and kept every 500 samples, resulting in 2,000 sampled ARGs. So here I was highly cautious by discarding 1 million of 2 million, but hopefully it would be about 20% of all in practice. So first, let's check the convergence. Here, the left plot is the trace plot of the posterior, and the right is the autocorrelation. So I've checked the convergence by the trace plot. So we see here that it demonstrates that the algorithm mixed well. I've also had independent run, and they all converge to the same place. On the right, the autocorrelation also, we see that from the first lag, the autocorrelation dies off to about 0.3 and very quickly goes to the 95% interval. And here I've also reported the effective sample size, which is 1,311 for 2,000 samples, which is very reasonable. And then now we are confident that the sample we got is valid for inference, and we proceed with the inference. I've also applied ARGweaver to all these 150 datasets, and here the CPU time is reported. For 2 million iterations of ARGinfer, for ratio 1, we need 19 hours, but ARGweaver for 20,000 iterations requires 5 hours. And for ratio 2, it's 6.5 and 4 for ARGweaver. And ratio 4 is kind of similar, but in general, ARGweaver is faster than our method. The first parameter we investigate here is the total branch length per site, which is in generation. So I've randomly subsampled 50 out of 150 datasets and shown here. The x-axis is the true branch length, and y-axis is inferred. The blue color is ARGinfer, and yellow is ARGweaver, and the bars are 50% credible interval. So we expect like 50% of them covers the truth here. As we can see, they perform very similarly, and they're nicely floating around the truth. But we might, if we take a closer look here, we might see that the ARGweaver actually tends to have lower branch length than ARGinfer. Like we see for these couples here, and some of them here, but it's not it's not for all. Like for this tail here, we see that slightly ARGweaver is higher. I've also reported some statistics for these. So the first is RMSE, root mean square error. ARGinfer has smaller RMSE. The coverage is similar, and the average length, ARGweaver has a smaller average length. So this coverage and average length are for 50% credible intervals. The next parameter we investigated is the number of ancestral recombination. So I should note that we ArgInfer can infer all the recombination events, including ancestral and non-ancestral, but Argweaver only infers ancestral recombination. So I've only reported the ancestral recombination here. Again, 50 datasets are randomly selected from 150 datasets and shown here. Similarly, x-axis is the true, y-axis is the inferred for both ArgInfer and Argweaver with 50% credible interval. So again, we see that they perform very, very similarly and floating around the true value. But as we go up here, we see that Argweaver tends to have smaller number of recombination than ArgInfer. So for this couple here, almost all these four. But again, it's not the case for all of them. Like here, we see that they're kind of equal. So the statistics, our method is better in terms of RMSE, and Argweaver has 0.49 coverage, 50% coverage, and our method has 0.57. So Argweaver does better. And in terms of average length, again, Argweaver does better slightly. So these are not significant. I've checked them, none of them, like RMSE for hours or the average length for them is not significantly different. So now let's go back to this question. So we see that there is a slightly different between the number of recombination, or slightly was different between the total branch length between ours and Argweaver. So which one is more accurate? We can see from this plot and answer that question. So this is the recombination rate estimation for ratio 1, 2, and 4 for all, so here for all 150 datasets. For ratio 1, so the red is the truth, and green is ArgInfer, and bluish is Argweaver. So we see that ArgInfer can infer the recombination rate with high accuracy. It's like the mean is exactly around the true value. On the other hand, Argweaver underestimates the recombination rate, and the mean actually is significantly different from the true value. I've tested them. The p-value for our method is 0.4, and the p-value for Argweaver is very tiny. So we can say that the inference here is biased for ratio 1. So let's check ratio 2. So ratio is mutation to recombination rate. Again, our method does very well, and the accuracy is very high, as we see. But so Argweaver does better in this ratio than ratio 1, which is expected because we have lower recombination rate. That means we have higher information about recombination, and the model can do better. But still, the mean is significantly different from the truth, with the p-values here for ours is 0.82, and for Argweaver a very, very tiny p-value. For ratio 4, our method is very consistent in this. Is again, we see that the true value is like overlapping with the mean. For ours, Argweaver does better than before, and the p-values are 0.55 for ours and 0.0139 for Argweaver. Okay, so the next parameter that we investigated is allele age. So what is allele age? So we have this the genealogies, and then we can map the mutation on the ARG, So a mutation can be on a branch. A branch has a starting time and an end time. We take the middle point of the branch as the allele age. And this graph is just for one dataset randomly chosen out of 150 datasets. The left graph is plot is ARGinfer, and the right is ARGweaver. And red is the truth, and shade is 50% credible interval, and the blue line is the inferred. So we see that both algorithms kind of did well in finding the pattern of the allele age, but like in this area, we see that ARGinfer kind of does well in following the truth. On the other hand, we see that ARGweaver is kind of going in another path. And according to the statistics here, for almost all the measures, RMSE, coverage, average length, and so Pearson coefficient is the correlation. For all of them, ARGweaver does better. ARGinfer does better, sorry. So we see that our method is more accurate for this parameter. And the last parameter we talk about here is the TMRCA along the genome. So again, one data set is randomly chosen. Here, x-axis is the genomic position, y-axis is the TMRCA. And so both models could kind of find the pattern of the true value very nicely, we can see. And the shade is 50% credible interval, and we see that it's very smooth for hours, but on the other hand, it's kind of this box-like for ARGweaver. And the reason is because ARGweaver approximates the time and gives some pre-specified discrete access for time, and the result is here. So the statistics again reported for this parameter as well. RMSE for our method ARGinfer is better. The coverage is similar. The average length again, our method does better. And the correlation slightly better than ARGweaver. So these were on simulation data. For real data, unfortunately, I don't have an analysis at the moment. There are some limitations that need to be addressed, one of which is the mutation model. So we assume infinite size model as a mutation that cannot handle back mutations and triallelic SNPs. So I'm working on adopting a more realistic mutation model that can handle these two. In conclusion, we developed an MCMC algorithm to perform inference under the coalescent with the combination. Our method assumes a more accurate model than ARGweaver and models non-ancestral recombinations. ARGinfer is more accurate than ARGweaver, but ARGweaver is faster. This is a novel method, and it can be improved to achieve greater efficiencies and tackle larger datasets. At the end, I'd like to thank all the people who supported me during my PhD and during my life. Firstly, my great supervisors, Professor David Balding and Dr. Yaoban Chan. I've been very lucky to be your students. Thank you very much for all your support and motivations and all your advices. I appreciate everything that you've done for me. I'd like to thank my committee chair, Dr. Nathan Ross. Thanks for all the positive and useful feedbacks you gave to me. And also I'd like to thank my collaborators, Dr. Jerry Koskela from University of Warwick and Dr. Jerome Kelleher from University of Oxford. So the collaboration goes back to last year that we could travel abroad. So I gave a talk in the ProbGen conference in France, and they heard me there. it turned out that they have the same idea on using TS for inferring ARG. So we had very useful discussions after my visit to Oxford last year. And then we established a collaboration that is continuing now. I'd like to thank Melbourne Integrative Genomics, my family in Melbourne, all the members, all the PhD students there. Thank you very much. I'm very proud that I'm a part of MIG. I also like to thank a School of Mathematics and Statistics that made this PhD possible for me, and Melbourne Research Scholarship and Shingley Scholarship for supporting me financially. Shingley Scholarship is a top-up scholarship offered by School of Maths and Stats. And I'd like to thank my family. I know it's been very hard. I've been far from you. I kind of broke the tradition of the family. Thank you for all your emotional support, and thank you very much for everything. I love you. And also like to thank my friends, my special friends in Melbourne and in Iran. And thank you for supporting me. And thank you all for listening to me. I'd be happy to take your questions and comments.
Well, thanks very much, Ali, for a very clear presentation. And I think you've explained all of that very well. And I would like to invite questions. As I said before, I think at the beginning, some people might be a bit scared to ask questions, knowing that he's a candidate for an examination. But as I explained, only good can come out of these questions because you can help suggest things. Feel free to type a question in the chat. There are none there yet, but if anyone would like to unmute themselves and ask a question, please go ahead.
This is Mike Goddard, if I may ask a question. You possibly did explain it, but I didn't quite get what the connection between the tree sequence method of storing the data and your MCMC method is.
Yes, thanks for the question. So the idea of the tree sequence is sharing, is storing like identical subtrees of marginal trees only once. So we use that idea and we accommodate that in our data structure. And then, so that helps us in the likelihood evaluation of not repeating, you know, calculation for every tree.
Thank you.
Other questions? It will be good, as I said, to you know, if there were any other unclear things, please, yeah, please go ahead and ask.
David, I put mine in the chat, but obvious question. Ali, I really enjoyed that talk. It was fabulous. An obvious question: how does this scale to realistic data sets? How does it scale up?
So for now, I assume it's a small data set, like let's say 10 sequences with about like 200 to 300,000 base pairs. But definitely I have ideas for improving the scale.
Do you have a sense of how, like you said, it's clearly not going to be linear, you know, are you going to be able to get to very big size data sets with this kind of method?
Probably not, but I'm thinking of the same scale as ARGweaver, like having like less than 100 sequences with 1 to 2 million base pairs. But definitely it can't go to, you know, thousands of sequences.
Hey Ali, thanks very much for the fantastic talk. It's been really great to see how this work has changed as you've done more and more work on it over the years. Wondering if you can give us a bit more explanation about those 6 different steps, or just 6 different types of changes in your MCMC sampler, and what some of the intuition behind why you've introduced all of them is. So like some of those changes, like, you know, resampling the event times, for example, seems like a relatively small change in that it doesn't change the topology of the ARG at all, whereas resimulating a whole subtree underneath an ARG is a really kind of nuclear option that might change a lot. Is there a, you know, are you, yeah, what was the reason for all of those different types of steps?
Yeah, so each of those transitions have their own purpose. Like, so let me go back to them. This is a very good question. Thanks for asking. So here, so here for removing, so we want to kind of move through dimension. So if we take the number of branches as dimension of the space, then when we remove it, then we reduce that. So that helps in kind of mixing, exploring the space, and adding is is expected, because when we remove, we need to propose an adding add a recombination. And this time, this one is doing all of them together, but in another scale. So let's explain add first. So for add, we choose one lineage and then randomly choose one to rejoin that is somewhere, and that result of maybe lower acceptance. But if the acceptance happened, then that would be a big change on the ARG. But the fourth one has focus on a local, like time interval by time interval, but it can kind of remove the restriction of if we had a recombination that needs to be removed or a recombination that needs to be added on a local of time interval window of the time. So kind of all of them work to help the MCMC mix better.
Can I just check I've got the right intuition, Ali? So
yes,
when you do the normal kind of removing a lineage and joining it back on, you are always going to the root in the simple version of that. But this re-simulating part allows you to rejoin elsewhere and move to a different part of the space efficiently. Is that right?
No, the SPR can, you know, we can choose any of the sequences, so it not necessarily goes to the root, but maybe the chosen sequence to rejoin would be like far from the pruned lineage on other subtrees. But when we do the resimulating, then we start from the pruned and going back time interval by time interval, and you know, shuffle the ancestral material for the sequences that are around there. So this also can resimulate the whole ARG, but it mostly are like in a window of the times. Please say if it's not clear, I can explain more.
Okay,
that was good.
Sorry, go on.
I was just going to say that was good.
Yeah, good, thanks. Any other questions for Ali?
I had a question. This is Ashley. Great talk, Ali. Really well done. I was wondering about inference, notwithstanding your comment, your answer to Stephen's question about scale. Is it fair to say that, well, you showed, but is it fair to say that this will perform better at deeper timescales? And have you thought about human data where we have this problem of very short, ancient, archaic haplotypes and trying to infer the age depth of those short regions?
No, actually, I haven't thought about that. Yeah, for real data sets, kind of, I'm still working on it. I have some, as I said, I have some other like limitations to need to address. So in short, no, I haven't, but I'd be happy to discuss later if you have any ideas to improve the method.
Thank you.
Hey, Ali. Hello.
Hi.
Yeah, there was a slide where you talk about estimating the recombination rate, and then you
Yes.
present some p-values. I was just wondering how you compute those p-values.
So the p-value is for each is against the truth. It's a t-test, both-sided t-test.
And how did you make those? Is it related to the box plot that you have there? No.
It is related to, you know, for each data set, we have a recombination rate estimation for each method. So we have 150 data sets, so we have 150 estimates for one method. And we also have the truth for the recombination rate. So what I've done is a t-test that if the mean of these are coming from the truth, I mean, are significant, are equal to truth or not. So that both side t-tests.
I see. So in practice, we actually cannot compute the p-values because we don't know the truth, right?
Yes. In reality, if we knew, then we don't need to estimate.
Right. So it's just like an illustration. Okay, I see. Thanks.
Yeah. All right.
To the end of the hour, so I'd just like to say, and I'm sure Yoban will join me to say thank you. Well, first, thank you again for very nice and clear presentation. Thank you for being such a talented and hardworking and good-natured student, and your contribution to MIG over the years. To everyone, Ali is going to submit it, we hope, in about 4 to 6 weeks from now. So if you have any further ideas, encouragement, suggestions in the next couple of days or weeks, please pass them on. And otherwise, thanks everyone for coming, and bye-bye.
Thank you so much, David.